DNA replication is the process by which DNA makes a copy of itself during cell division. To fully understand the process, we should start with the enzymes that are involved in the process of DNA replication. Topoisomerases are enzymes that participate in the overwinding or underwinding of DNA. They initially ease the tension between the DNA bonds. A DNA helicase is an enzyme that functions by melting the hydrogen bonds that hold the DNA into the double helix structure. During DNA replication, SSB molecules bind to the newly separated individual DNA strands, keeping the strands separated by holding them in place so that each strand can serve as a template for new DNA synthesis. Primase is an enzyme that creates a primer on a DNA strand by adding RNA nucleotides to the strand according to the DNA template sequence. Primer is a short nucleic acid sequence that provides a starting point for DNA synthesis. DNA polymerase III adds nucleotides to the 3' end of a DNA strand, one nucleotide at a time. DNA polymerase I is essential for removing of the RNA primers from the fragments and replacing it with the required nucleotides. DNA ligase is an enzyme that repairs irregularities or breaks in the backbone of double-stranded DNA molecules. The topoisomerase usually comes along with the helicase as they work together to break down the DNA double helix to initiate the replication process. The topoisomerase breaks the phosphodiester bonds of the DNA, while the helicase breaks the hydrogen bonds of the DNA, thus finalizing the unzipping process. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds, holding the complementary bases of DNA together, A with T, C with G. The separation of the two single strands of DNA creates a Y shape called a replication fork. The two separated strands will act as templates for making the new strands of DNA. Following the unzipping is the appearance of the SSB proteins to aid in separating the two strands, preventing them from reconciling again. One of the strands is oriented in the 3' to 5' direction towards the replication fork. This is the leading strand. The other strand is oriented in the 5' to 3' direction away from the replication fork. This is the lagging strand. As a result of their different orientations, the two strands are replicated differently. A piece of RNA called a primer, produced by an enzyme called primase, comes along and binds to the end of the leading strand. The primer acts as the starting point for DNA synthesis. DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide bases A, C, G, and T to the strand of DNA in the 5' to 3' direction. Then comes the DNA polymerase I to alter the primase and turn them into nucleotides. And lastly, the ligase appears to combine the fragments of nucleotides in the strand. Numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called Okazaki fragments are then added to the lagging strand, also in the 5' to 3' direction, through the usage of the DNA polymerase III. The DNA polymerase I then swoops in to alter the primer once again and make them nucleotides. To end the first segment of the cycle, the ligase appears to patch up things in order and stands by to patch up the following segments.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around the proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm called ribosomes bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different amino acids. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
To give an example, to create the protein insulin, it is necessary to synthesize 21 amino acids for the A chain, while the B chain must have 30, a total of 51 amino acids needed to create insulin.